called Plasticity and Constancy, Philosophical and Scientific Perspectives. And it's my honor to present you the first speaker of this session, Professor Ute Deichmann. Ute is a professor of history and philosophy of science here at the Ben-Gurion University of the Negev. And she's also the founding director of the Jablov Center for the History and Philosophy of the Life Sciences. She's the author of Biologists Under Hitler from Harvard University Press, editor of many books and journal volumes, and author of tens of articles and book chapters. Professor Deichmann has a PhD in genetics from the University of Köln and held academic positions at the University of Köln, Leo Baeck Institute London, and the Hebrew University of Jerusalem. So, join me in welcoming Professor Deichmann.
I don't have a PhD in genetics. I have a PhD in the genetics department because they were unable to give me a PhD for history of science. Then they would have to give me tenure in that one. Okay. Yes, my lecture is actually almost the lecture, the title of the whole workshop. No wonder, because I thought about it. The idea of constancy in development and evolution, scientific and philosophical perspectives. So, a short introduction. The antinomy between plasticity or change and constancy or plasticity was a part of many myths about the creation of the world. For example, in the Torah, in the Bible, we read that instability and formlessness were first before the organized beings were created one by one. It was also, this antinomy was also a major topos in European history of thought since pre-Socratic times, in particular in disputes about Parmenides' ontology, which said that constancy prevailed and change was only apparent. And third, this antinomy is, as we have heard in several lectures today already, an important topic in modern biology, particularly developmental biology and evolution. That means the real biological stuff, not the physiology and so on. That's at least how it is perceived. So, coming to biology, change is everywhere. Random processes, for example, thermal molecular movements, Brownian motion, are ubiquitous. They, on the one hand, enable biochemical reactions and have led to the diversity of organisms, to biological specificity and the individuality of organisms. Otherwise, we would be all clones of one another. And second, they also destroy order in life by increasing stochasticity and by preventing deterministic outcomes. So, on this basis, it is in a way a miracle that there is stability. We have species stability over long periods of time, as we certainly Doug Irwin will tell us tomorrow about, and we have the constant outcome of development. The French biologist and philosopher Jean Rostand wrote, "Theories pass, the frog remains." And this is a sentence that François Jacob included as a motto in one of his books, the molecular biologist. It is usually interpreted as theories can be discarded, but the facts remain. I venture a new interpretation. Namely, good theories have to account for the fact that the frog remains a frog in the next generation and has remained a frog for more than 50 million of years. And this developmental and evolutionary stability is related, as we know now, to properties in the system of transgenerational information processing, about which we have already heard and will hear in other lectures. And thus my goals here are, I claim that the greatest challenge to understanding development and the history of life is not the change of species or plasticity, but stability and constancy. And second, I argue that explanations for the constancy of developmental outcome and the stability of species, based on Davidson's idea of the regulatory genome and the many predecessors he has had, are superior to those based on purely physical methods, though physical models are important, as we heard in James' lecture. And third, I propose, and that may be quite a chutzpah, but I continue it, I propose a parallel between modern biology and ancient Greek philosophy in the one sense, namely in the idea of change being based on constancy. And I will explain in a moment. So I have 3 points. First, the purely historical point, the generation of the idea of constancy in natural history. Then I briefly touch different explanations of stability of development and the constancy of species. And third, I talk about these parallels between ancient philosophical thought and modern biology. First point, the generation of the idea of constancy in natural history. It is widely believed that prior to the 19th century, Darwin, before that Lamarck, the Christian doctrine of creation was the dominant view of species diversity, and that it was reinforced by ancient Greek texts of essentialism and typology, especially, of course, by texts by Plato and Aristotle. It is also believed that Darwin's major achievement was to overthrow ancient Christian beliefs in species stability. However, this is not correct. The fixity of species was not an ancient Christian belief. I can talk about it, but I don't have the time. And we can see that the ideas of species change or the transmutation of species and of spontaneous generation was widespread until the 18th century, at least, and in some cases until the beginning of the 20th century. We have, on the one hand, the generation of species from decaying matter, or from inanimate matter even, especially of small animals. Then there are popular notions of monsters that blended characteristics of existing species, which appeared also in textbooks of zoology, like this creature here, which is called Lamia. And we have the belief that transmutation, change of species, happens through hybridization, and that environmental influences are inherited. So this is widespread ideas. And it was Carolus Linnaeus, or Carl von Linné, the great Swedish botanist, who first proposed and defended and promoted the idea of a quasi-species fixism. He was not so strict on that either, but still, species got specificity with him. And he also proposed the first comprehensive taxonomy based on the hierarchy of beings, a taxonomy which has been amended and changed by himself and many others. And here we have a section of it, namely the animal kingdom, and the kingdoms are listed here somewhere, I can't read it, under quadrupedia. So humans were involved in this taxonomy. This classification, which presupposed the long-term stability of traits, was not, as many believe, an obstacle to the development of evolutionary theory, but on the contrary, it was a prerequisite for generating an evolutionary history of the living world. And I quote here Michel Moran, whom I also quoted in the introduction this morning, a philosopher of science, a philosopher of biology, to be concrete, who wrote that the theory of evolution has no place in a world where the existence of monstrous hybrids was accepted or where the most fantastical transformations were anyhow conceivable. So this transition from concepts of fluidity to those of constancy and specificity in the 19th century took also place in many other fields. And it actually was a prerequisite for biology to become a science and not just a descriptive enterprise. So we have it here, for example, in cell theory. We have it concerning the individuality and specificity of bacteria, an idea of Ferdinand Kohn, on which then Pasteur and Koch established the germ theory of disease. And we have it in genetics, where with Mendel on the one hand and Boveri and Wilson on the other, stable concepts were introduced, namely that of elements, which later became genes, and of chromosomes, which were had an individuality. So, and yeah, I said this, and this was, all of this stability was also the prerequisite for the scientific study of change, for example, mutation, recombination, and so on. And now I come to my second point. explanations of stability and development, and species stability. Sorry, it's missing. So, first, genetic and genomic approaches. After 1900, when Mendel's works were rediscovered and formal genetics started to thrive, several scientists, among them Moran in Germany and Sutton and Morgan in the United States, proposed the chromosome theory of heredity, which says that genes on chromosomes are the basis for heredity and development. And it was a good idea because chromosomes were stable, genes were stable, and they could be located on chromosomes by Morgan and his school. You see here the results of many mutation experiments which they carried out for many years. So now we have stable elements in biology, but the question, of course, remains, and that is a question we have talked about or heard about often this morning, and that was raised in this case by Jacques Loeb, after whom I named this center, that what molds these independent hereditary characters into a harmonious whole. Nice, they exist, but what to do with them? And we know, of course, now that it is a question of regulation, and it is interesting that it took decades of time until the first hypothesis, based on experiments, were proposed. Jacob and Monod, the Opron model is one of them. And so that about 100 years after Loeb, Eric Davidson could still say no observations on single genes can ever illuminate the overall mechanisms of development of the body plans or of body parts, because they disregard the holistic processes of development. I say this here for people who are not so familiar with this very briefly. Davidson introduced the concept of hierarchical gene regulatory networks, to which Ed Halper in the morning referred so often. And we see here one of his books, in 2006, The Regulatory Genome. And the networks here, a short section of it, consists of regulatory genes which regulate other regulatory genes, and so on, until at the end we have genes which code for proteins, and they are the actual let's say, servants of the body. They make what we can see. They catalyze, they build, and so on. So when I say these networks control development, I want to make a short digression, namely the phenomenon of phenotypic plasticity. That means the ability of a genotype to generate more than one phenotype in response usually to short-term environmental changes, which happens particularly in insects. And I like these creatures here very much, maybe because I now live in the desert. This is the desert locust. And this is a very benign creature. Green, it lives solitary, it doesn't do any harm to human crops. But when under certain environmental conditions, especially under drought conditions, they change. That was a result. They change not only their color, their phenotype, they become colorful, black, red, yellow, and so on, but they also change their behavior. They become gregarious, and they fly around in swarms and destroy what they can of human crops. And we recently read the Haggadah, the plagues against Egypt, and we have here one of the swarms, and they don't leave much behind. So when I say the genome is, or Eric says the genome is controlling development, that means the genome does not control every development trait, but it fully determines the range of potentially reversible phenotypic variations. That means the reaction norm. Yeah, Eric Davidson, together with Ben-Gurion, he sits here somewhere. They are among the They are, and you have to correct me, but I think you are among the very few people who, in the whole of Evo scientists, are also proposing hypotheses for a causal mechanistic explanation of evolutionary stasis, not only of evolutionary state change. And these are just two of the papers they wrote together. And in this evolutionary stasis, the long-term stability of species, until in the end they change, most of them at least, is built on the stability of developmental outcomes. I said before, in the introduction, the dog begets the dog, etc., etc. And this is, at least according to them, behind the evolutionary stasis. And they also explained the extreme conservation of certain morphological features over immense geological periods, in part by the hierarchy of gene regulatory structures. So, I cannot really show you this, but you can imagine that these gene regulatory structures, they are, as I said, hierarchical, and we have at the periphery genes. When they are changed, the viability of the body of the organism is usually not affected. They can increase the viability of species, the variability of species, for example, or also of organisms within a species, but they don't change the viability. But then we have changes in the center, and somebody mentioned today kernels. And when there is a change, a mutation or something, that affects the body very, very severely, because it can affect the body structure or some vital functions. And in that case, the organism doesn't survive, and that means they don't exist. So we have a stability based on the non-viability of changes in the kernel. I'm pretty sure that Doug will talk tomorrow about these kernels and of a different view they meanwhile develop, but I mean, this is what I know. So, Eric's developmental gene regulatory network model has been challenged by, of course, by new research and is being transformed accordingly. But I think that the ideas of the causal role of these networks for development and their hierarchical organization have been confirmed in numerous different studies and from numerous different animals. And they have remained most fruitful for the explanation of the stability of development and of evolutionary stasis. Yeah, I switch now briefly to physical chemical models and focus on reaction diffusion models, which were introduced into biology by Alan Turing during his research on brain development in 1952. And Turing introduced these reaction diffusion equations to explain how spatial patterns in an egg form autonomously. And the idea was that two homogeneously distributed substances with different properties, they spontaneously generate inhomogeneous patterns of the substances in the egg, and he called them morphogens. James, that's why I'm so interested in these morphogens. I think you need something a little bit different. And thus produce novel shapes and gradients. And here are some of the shapes that can be produced on the basis of these equations. So these models have been tried to, by biologists, to explain, especially Drosophila development of the stripes on the Drosophila back, but it was not very successful. And updated Turing models with more than two factors, etc., they were used to simulate the development of periodic patterns, like here the beautiful models to simulate stripes in fish, various fish, and here Hans Meiner's patterns on seashells, which are very beautiful. They look almost like, or they do look like the real seashells. And he, I think, devoted decades of his life on modeling them. He was a mathematician. The problem is that these simulations, they don't reflect reality. They didn't do what James was doing to conduct at the same time also experimental perturbation experiments. And the problem is that they neglect the already formation in the egg, in the unfertilized egg. It is not an homogeneous stuff there. They are not organism-specific, and genes are excluded, since Turing are excluded from the discussion. And so Sean Carroll wrote, the evolutionary biologist, wrote, the mathematicians never envisioned that modular genetic switches held the key to pattern formation, and that the periodic patterns we see are actually the composition of numerous individual elements, like these beautiful stripes on the Drosophila back. An early paper which already pointed in this direction is by Michael Akan, and I think Ariel, you're a student of him, right? Okay. Yeah, so 1989. And he showed on the basis of his own experiments in Drosophila that elegant mechanisms, like those of Meinhardt, do not generate the periodicity in the repeating stripes in development. And he wrote, the apparent simplicity of the repeating segment pattern might be deceptive. It seems that spontaneous pattern generation mechanisms do not define the periodicity. If they do anything, they just sharpen the boundaries between the stripes, but they don't create them. And so
his views changed following that.
Okay, let's talk about that. That's what he wrote, but yeah. So anyhow, if this is the case, if this is not the case, we will hear it later. So I asked, why do Drosophila and other organisms not use reaction diffusion mechanisms to generate their signals? And one hypothesis in this, to answer this, is the less elegant mechanisms are more robust and therefore favored evolution. And this has been proposed by systems biologists, for example, Kitano, who wrote in a very often or frequently cited paper that robustness facilitates evolvability, and robust traits are often selected over non-robust ones. There's another problem, because how is it that evolution does not only lead to robustness, and we look at the viruses, which are a product of evolution, but very much not robust, as we know with this COVID stuff. And some robust organisms hardly evolved at all. For example, living fossils, here the limulus creature. I understand there are meanwhile also other interpretations, but they really seem hardly to evolve, and they are very robust. So another point here is that gene networks contain robust and not robust elements. So robustness doesn't seem to be an absolute value, but it is necessary for longer-lived organisms, independently of evolution. And the major problem with these robustness models is that, to my knowledge so far, there are no detailed mechanistic analysis. So, a third point, recently, and I find it most interesting, and you can see where that leads to, these physical-chemical mechanisms are integrated into genome-based models, starting here in the generation of certain periodic patterns in animals. The first example is a paper by Sharp et al., which was also very, very often cited in 2014. And they gave evidence that digit specification is driven by a Turing network that is implemented by gene products. ENP, I think, is a morphogen, SOX9 is a transcription factor, and WINT is a signaling pathway, gene, I mean, to signaling pathway. And we have here on the right side in experimental results, and here on the left side the modeling. There are quite nice overlaps. So I was very impressed until recently I talked to a colleague in Köln, Preformation, and he told me this is mainly a simulation experiment informed by some experimental input. But the problem is that the basic assumptions are highly speculative. And he explained to me that equations like the yellow one, where you have the transcription factor SOX9 with an exponent of 3, this is very important because only then can you have non-linearity and create spatial patterns. But there is no whatever data to support this, which is purely mathematical component that is required to create the spatiality. So it seems also that this beautiful model is not empirically verified, at least not at the time being. Second example is by Economy et al. Last 3 years ago. They conducted a perturbation analysis of the Turing-like reaction diffusion stripe patterning system of ridges in the mammalian palate in the throat and the regulatory interactions involved in this process. And also they show by experiment that there are many factors cooperating in these stripes in the throat. And they point, interestingly, this goes back to, supports Akan, so I wonder what to say, points to discrepancies between the results of mathematical models and biological reality. So why does the patterning, for example, of the palate use 5 pathways in the organism when, in principle, 2 would do to reach the same results in the models? And they discuss 2 possibilities, so this is both speculative, that there may be a function for these pathways, maybe they provide for more robustness. But there may be also that evolution brings forth non-functional structures or processes. This is reminiscent in my mind to the paper of Gold and Leontine of 1979, I think, the Spaniels of San Marco, in which they showed that there are many structures created by evolution which don't have any particular function, but they have to be there for architectural constraints, developmental constraints, and so on. And they say, so it could be that all the regulatory interactions are used elsewhere for multiple other purposes, and that the apparent overkill in terms of numbers of pathways involved is merely because there is no evolutionary pressure to eliminate or suppress the role in the palate. And they conclude that patterning is brought about by non-elegant processes like Michael Akan. And I like this in a way because it reflects Francis Crick's dictum that elegance and simplicity are in biology dangerous guides to the correct answer, because the organisms are not designed, they are evolved. So, my last point. How much time?
5 more minutes.
Okay, great. Change based on constancy, parallels between ancient philosophical thought and modern biology. An ancient debate in developmental biology or developmental philosophy since ancient times was about preformation and epigenesis. Preformation, the embryo or parts of it are preformed in the egg cell. And we can also subsume Hippocrates under that, who assumed that the structure of the adult is present in the germ cell because every part of the adult brings parts of it to the germ cells so that it will resemble the next generation. And the opposite opinion is epigenesis, that embryonic development starts from homogeneity, and the complexity increases only over time. Many reasons, materialistic reasons, immaterial reasons. So that depends on when and by whom you look at. So at around 1900, the American cell biologist Edwin Wilson proposed a synthesis, and I find it very, very interesting and far-reaching. He said the apparent cytoplasmic epigenesis, that means increasing complexity and the ability to regulate of the embryo, is based on the transmission of a nuclear preformation. We would today say on the DNA in the nucleus, but he wouldn't know it then. So, now coming to philosophy. The debate over preformation and epigenesis is also a debate over continuity or stability and change of plasticity when we look at it in an abstract way. The British classicist G.R. Lloyd wrote that the problem of change became the chief problem in philosophical thought about nature in the 5th century BCE. Changes seem to occur, but do the appearances correspond to the underlying reality? So we have Heraclitus on the one hand who emphasized changes, at least that's what we mostly believe, and Parmenides who proposed his philosophy of being, that the multiplicity of existing things with their changing form and motion are only an appearance of a single eternal reality, which is the being. And Lloyd says, and that I found so interesting, and that's actually why I proposed this workshop, said that the most important philosophers of nature of the late 5th century took their starting point from Parmenides. So we have Empedocles, who proposed the physical elements, he called them differently, elements, the term of Plato, I understand, but it doesn't matter, as roots, he called them roots. As original substances that have existed as long as anything has existed. And they produce change by mixing with each other, by separating from one another, and so on. But this, so change can occur, but the substances, the elements themselves, are unchangeable, as Parmenides' unchanging being. And similarly, Democritus, the ancient atomist, who believes that atoms of the void are alone are real, they produce change through the combination and separation. But atoms, again, are indivisible and eternal, as Parmenides being. So what we have here is the suggestion of change based on constancy. And I think that the notions of the cell nucleus, coming back to Wilson, as an original preformation, and heredity as being affected by the transmission of this preformation, which in the course of development is expressed in cytoplasmic epigenesis, shows parallels with the arguments of Empedocles and the ancient atomists. They are changes based on the movements of unchangeable, pre-existing entities. I need a few more minutes. So, I now turn to Schrdinger, whom I also cited before, and who asked in his Nature of the Greeks, how did the ancient atomists arrive at conclusions that in many ways are similar to modern atomism without any empirical evidence? And he continues to ask, is the thought pattern in question perhaps not so exclusively based on the recently discovered evidence as the most modern thinkers believe, but on the cooperation of much simpler facts known before and on an a priori structure, or at least the natural inclination of the human intellect? If we take this seriously and try to transfer to biology, we have to ask what were the either conscious or maybe unconscious observations made by both ancient philosophers and modern biologists in regard to development. And certainly we arrive at least at two answers, namely its constant outcome and the similarity of parents and offspring. And so when we take this and take the elements and the atoms and the genes and chromosomes, the problem still remains. If elements, atoms and genes are the source of this constancy, how is their activity organized? So we reach a point which Ed Halper raised already and came also in other lectures. And here I want to have a short digression on Max Delbruck. James, you mentioned him in the last slide. He was a theoretical physicist who turned to biology and became one of the founders of molecular biology. And he wrote an article, Aristotle, in a Festus, I don't know an English translation for that, for his French colleague, André Lwoff, both of whom received a Nobel Prize for molecular biology. And he wrote, during the past 20 years, André Lwoff, mon cher ami et collègue, and I have been carrying on a casual correspondence about Aristotle. I was very intrigued when I wrote it. I mean, the whole article is not so seriously, though it contains serious elements, but in the 1970s, famous molecular biologists still read ancient texts. And I actually spent a large time of my life in an institute founded by Edwin, and the first generation of geneticists were also reading ancient texts, even part of them, some of them even in Greek. And so I like to read this. And so now there is dictum, and don't take it so seriously. So he wrote, Aristotle discovered some 2,300 years ago DNA as carrier of genetic information. Aristotle said, this is a quote now, from their works, Aristotle, but he took it from the Generation of Animals, first book of it, that the male contributes the plan of the development and the female the substrate. Let's leave alone the sexism, but focus on the plan. So he continued, the male semen is thus an immaterial principle of form that does not contribute anything to the material body of the embryo, but only communicates to it its developmental program. And this principle of form is the information stored in the semen, which after fertilization is read out in a pre-programmed way, the genetic program. What I said today morning in a comment, the problem is that the principle of form today is a material one, and that we cannot separate immaterial information from material molecules. And also that the principle of form is not unchangeable. And to quote again Michel Moran, the secret of life resides in the systemic relationships supported by certain types of molecules in the organisms. The living world is therefore the product, both structurally and functionally, of a particular kind of chemistry, and matter and form are completely intertwined. To summarize, one last part. Constancy in nature is based on organism structural and organizational hierarchies and genetic causality, as I see them as fundamental principles of life. The genome plays here a prominent role in generating constancy in development and evolution. But unlike the atoms and elements in antiquity, genomes are highly structured. They are hierarchically organized and play a causal role in development. And that is why they can be the carriers of constancy, of form, and organized change at the same time. I thank you.
Thank you, Ute. So we have 6 minutes for questions.
I have 2 comments related to 2 different parts of your talk. So not questions, but comments. I'm going to monologue a bit. Fill up for me. So one, going back to Michael Akan. When he was writing, he was talking about Drosophila. And in '89, that's pretty much the only arthropod model that was known, and Drosophila makes stripes inelegantly. But then work in his lab in the late 90s and early 2000s looked at many other arthropods, and actually most arthropods make segments much more elegantly than Drosophila. So that whole argument of inelegant as a process, as a byproduct, falls apart when you look at the diversity of arthropods and not just at Drosophila. But maybe I'll talk about that in my talk. The second point has to do with the idea of preformation and Aristotle. So it's not just the genome, but it's also the ovum. Mm-hmm. And in that sense, Aristotle is right. Also the egg, the ovum. So Aristotle is actually right, because when you clone an organism, you have to take the genome and you have to put it into an ova of the correct species. Because there's a lot of information, a lot of preformation that's already in the unfertilized egg that's laid down by the female. And that's why cloning mammoths would never work. Even if you have the full genomic sequence of a mammoth, you need a mammoth egg to put the genome into. So the preformation, the modern interpretation of preformation that both Aristotle and Lewis, was it? The 1900 person you mentioned?
Yeah,
Wilson. So the preformation there is not just in the nucleus, but also in the egg itself, because there's a lot of early information, early patterning information that's in the egg, and it won't work if you don't have the correct egg. So the genome won't work if it's not in the correct egg.
To the last first. A lot of the information in the egg is a result of the mother's genome. It is messenger RNA and other
yes.
products of the genome. So you cannot use this as an argument against what I said. And second, I mean, when you use the experiments of Craig Venter, who just transplanted DNA and produced it in a different environment, exactly the same kind of phenotype of a bacterium. So it is the information in the egg, in the DNA. Sorry.
But in multicellular organisms, that's not enough. You also need the information that's in the egg, not just the information that's in the nucleus.
Yes, but as I said, the information in the egg is
Yeah, of course, it's encoded by the genome. Everything is encoded by the genome. But it's not the nucleus alone. It's the nucleus and the environment of the egg.
Yes, as a result of the nucleus of a different organism. That is not an argument. So please, I have to reply to the first one. And I talked to developmental geneticists, and they said in Drosophila it is not elegant. The stripes are not produced by in a Turing-like matter.
Right.
But they haven't succeeded to do that all the years and decades since Turing. So, and what I learned also is that the reaction diffusion system, it works, seems to work very properly in bacteria, for example, to explain and predict bacterial fission. Then that you can do with this model, but not yet with any higher organism. That's what I heard from them.
There's Denny, and then...
I very much enjoyed the talk and your analysis of some of the recent work. In fact, there were more empirical evidence in favor of preformation than in favor of Turing mechanism to make digits. As a matter of fact, if I'm thinking of the experiment of Charles Bonnet on aphids in the 18th century.
Yes, you're right.
But the point I wanted to raise is on this striping pattern in Drosophila. In fact, it's interesting to realize that Hans Meinhardt did not look at the system from an evolutionary viewpoint. In fact, if you would have done that, you would have understood that, in fact, what is important is any kind of mechanism you're thinking of, it has to accommodate an evolutionary transition. And if you look at short-chambered insects from which the Drosophila derive, it's extremely complicated to think of how the system would have evolved from the short-chambered to the long-chambered. In contrast, it's much easier to think that short-chambered insect started to make one band, and then two bands, and then three bands, and therefore that this mechanism is impossible. I mention this because I think, I don't like the citation from Dobianski, but in this particular case, I think talking about this mechanism without integrating them into an evolutionary thinking is actually useless.
It's exactly what?
It's useless. It's useless. You really need to think at the origin of the mechanism to understand whether it can work or not. And the Turing mechanism type for the bend could not possibly work if you think of the kind of animals that gave rise to Drosophila, the long-German insect.
I think also not from a developmental perspective, not only evolutionary.
Yeah, from the development perspective, you can always think that from the time point you create an open space, then you can think of a system where you may have interaction and so on. But that only if you think of if you take a snapshot and say, well, this is an open space. But if you think that this open space was not an open space at the origin, then it kills the system.
Yes, in principle you may be right. On the other hand, I talked to Jen also, when we have this diffusing substances in development, the development is characterized by a lot of movements of cells. And let them diffuse, and where will they end up in after the next movement? It is a very difficult conception for that, But anyhow, this can be only decided experimentally, and I'm open to everything. I wish they succeeded.
Final question from Stas.
No, from any of
Oh, Annie, okay. Stas and then Annie.
Yeah, so I want to bring up the example of a mammalian egg that does not contain any localized determinants, and where the first symmetry breakings involve a self-organized polarity formation. So you have cleavages that give rise to 8 cells that are organized as tightly packed ice cream cones. And there are polarity proteins that localize to the ice cream part and the lateral parts in the excluding way. So from there on, this is So this becomes a pre-patterned state on which later cleavages elaborate, giving you trophectoderm and inner cell mass. But the first step is quite self-organized, which can be proved by dissociating an 8-cell blastomeres, and they do this even in the spherical case.
It's not guided by the RNA in the cell.
No, not at all. Not at all. So there are no, in contrast to what is happening in frogs and in fish, and certainly in insects, there are no localized determinants. These embryos can be completely reshuffled. Yeah, so
Mammals are derived. No, I'm joking.
Sure.
But it's true. I mean, there's a very wide range, I think, of degrees of pre-localization in the egg, and we are at one of the extremes of very little pre-localization, if any, that can be detected.
Yeah.
Interesting. So we should be special, right?
Final question for Mammals?
Mammals. Yes.
Is there time? Maybe there is.
We started a bit later, so
Oh,
we took a bit.
I would just point out that for Aristotle, with one exception, which I'll talk about tomorrow, you never encounter form as something immaterial without matter. It's not, you never encounter form without matter. So it's not that form is an immaterial principle, matter is a material principle, and the two of them then somehow come together in organic beings. In fact, he says in metaphysics that in organic beings, the form and the matter are essentially indistinguishable from one another, because no matter how low you go from the whole organism to the organs, and then from the organs to the tissue, and then from the tissue, you will always encounter matter in some form. So you never encounter matter without form. That's an abstraction. It exists in thought, but not in reality.
But what about this distinction of the male and female element? That he separates them very clearly.
Yes, that's true. That's true. But each of them is itself a form-matter compound. He's, of course, incorrect on what they both provide. He thought that only the male provides the formal principle. That's obviously faulty, but there's no. It's not that the female is matter walking around without any form. It's a human being. It's just that there's a difference in the mechanism of embryology, but not in her being a form-matter compound.
Okay. I grew up in an environment in which the role of the female was very much related to this victim of Aristotle.
Well, that's true, but that's not Aristotle.
Thank you.